Hi, I'm John Alcorn. My laboratory is over at the Children's Hospital in Rangos Research Center in Lawrenceville. My lab is focused on influenza and bacterial pneumonia, mostly in mouse models, but also in translational samples from patients here at our Children's Hospital. My lab is a medium-sized lab, about 10 people, 4 graduate students currently, and we are really focused on complications associated with flu, the primary one being bacterial infection, secondary bacterial pneumonia caused by MRSA. We've been studying for about the last decade mechanisms by which flu makes you more susceptible to disease. We've focused a lot on how that's happening in terms of interferon production induced by the virus and how that opposes host defense against extracellular pathogens. We've worked a lot in recent years on understanding cell dysfunction in co-infections and polymicrobial infections, these being both macrophage, neutrophil, T cells that are deficient or not functioning properly as they would in a single bacterial infection. And then we've tested a lot of interventions over the last few years, both partnered with pharma, but also through our own funding with NIH to try to find ways to either modulate the host response or modulate bacterial pathogen virulence factors or clearance mechanisms. A lot of what we study is actually the immunopathology that's induced in the lung. Something that's common to the flu field is this idea of cytokine storm. And cytokine storm or exuberant host responses to the virus actually drive most of the injury. And this is something that we think is going on also with SARS-CoV-2. In the context of flu, people don't really die of viremia. They die of host-mediated pathology that basically eliminates lung function due to either edema or tissue damage. So we have several projects in the laboratory in this area as well, trying to understand how flu or co-infections are able to drive actually pathogenesis. In recent years, we've studied the inflammasome in this process, but also STAT1 and STAT2 mediated inflammatory pathways. We are also interested somewhat in influenza repair, and one of the graduate students in the lab is working on a repair project trying to understand how the lung comes back from a viral infection. More recently, one of the grad students in the lab is working on memory responses and how T-cell mediated memory against influenza may be strain or heterotypically protective across strains of influenza. So in those ways, we're trying to extend our findings in acute illness into kind of chronic outcomes following flu. Finally, then, recently we've been involved in a lot of CDC-supported clinical trials for influenza vaccines. In recent years, that's been in children. We've been testing different types of licensed vaccines, whether that's the flu shot or flu mist. Those networks then follow up in the flu seasons with a surveillance network of adults and in children, where we collect samples from patients with flu, and we can then go back and look at the memory responses or T-cell responses induced by influenza in natural infection versus what's induced by the vaccine. And those type of comparative studies allow us to maybe better design vaccines as natural infection induces more robust immunity. So that's a general overview of what we're doing over here at Children's. Hopefully if you're interested, feel free to send me an email and we can chat.